Good day, everyone. Today we'll be delving into a fascinating and fundamental concept in biochemistry, isomerism. So, this topic is crucial for understanding the diverse structures and functions of biomolecules within our bodies. By the end of this lesson, you will be able to achieve several key outcomes. First, you will understand isomerism and stereoisomerism, which includes defining isomerism, explaining its different types, and describing the importance of stereochemistry in biochemistry. Second, you will be able to recognize and differentiate optical isomers, understanding chirality and optical isomerism, and identifying chiral centers in biomolecules. Third, we will learn to classify D and L isomers, distinguishing between these forms in carbohydrates, amino acids, and other biomolecules, and using Fisker projections for classification. Fourth, you will be able to relate stereoisomerism to biological activity, analyzing its role in enzymatic reactions and metabolic pathways. And finally, you will apply your stereochemical knowledge to solve real-world problems, especially in health and medicine. Let's start with a basic definition of isomerization. Isomerization is the process by which a molecule rearranges its atoms to form a new compound with the same molecular formula but a different arrangement of atoms. This process results in isomers, which are compounds that share the same chemical formula but differ in structure. Now, this is essential for the creation of different forms of sugars, contributing to their diverse biological roles and chemical reactivity. Now, there are many types of isomerism, including stereoisomers, the D and L, epimerization or epimerism, functional aldose-ketose isomerism, and anomerization or anomerism.
Checkpoint.
Which isomer type affects 3D shape and function? Stereoisomers are the isomer type that affects a molecule's 3D shape and function, as they have the same molecular formula and connectivity, but differ in the spatial arrangement of atoms, leading to different physical and biological properties. Moving on to chirality and optical isomerism. Chiral molecules exhibit a left and right-handed nature, meaning they exist as mirror image forms that cannot be superimposed by rotation. These molecules interact with plane polarized light. Those that rotate the plane of polarized light to the right are called positive or dextrorotatory forms, while those that rotate to the left are called the negative or the levorotatory. Due to this property, chiral centers are also known as optical centers. An important concept here is racemic mixtures, which contain both enantiomers in equal proportions. These mixtures do not rotate polarized light because the effects of the enantiomers cancel each other out. You can see an example of chiral centers highlighted in the glucose molecule diagram on this slide. Let's elaborate on enantiomers. Enantiomers are non-superimposable mirror images that are identical in most properties, except for their interaction with plane polarized light. This interaction is why optical isomers are named as such, because of their effect on plane polarized light. Simple substances displaying optical isomerism exist as two isomers known as enantiomers. The presence of asymmetric carbon atoms is central to a compound's optical activity. Plane polarized light is light that vibrates only in one direction. A clockwise or rightward rotation of plane polarized light is termed D, or dextrorotatory, denoted by a positive sign, while a counterclockwise or leftward rotation is termed L, or levorotatory, denoted by a negative sign. This slide provides a visual aid for finding chirality centers. Chirality centers, also known as stereocenters, are crucial for understanding stereoisomerism. The attached video link provides a more in-depth explanation on how to identify these centers within molecular structures.
In this video, we're going to focus on finding chirality centers. So in this example, we have a chlorine atom, a bromine atom, and also a methyl group. So how many chiral carbons can we identify in that molecule? So a chiral carbon is basically a carbon atom with 4 different groups. Primary carbon atoms are not chiral. This one is a CH3, and so it has 3 hydrogens on it, so it can't be a chiral center because it can't have 4 different groups. Now this carbon with the chlorine atom is chiral. We have a methyl group on the left, a chlorine atom, an invisible hydrogen atom, and then this entire thing makes up the 4th group. So this is the first chiral center. Now we have another chiral center here, because this group, that's one group, the bromine makes the 2nd group, and this is the 3rd group, and the 4th group is the invisible hydrogen. Now what about this carbon? Is that a chiral center? Well, that carbon doesn't have 4 groups because it's attached to 2 methyl groups. So this is not a chiral center. So we have a total of 2 chiral centers. So how many possible stereoisomers can we get for this molecule? The number of stereoisomers that can be produced is 2 to the n. So it's 2 squared, in this case is going to be 4. Now what about this example? How many chiral centers can you identify in this molecule? So is this carbon a chiral center? It turns out that it is. We have a methyl group, a hydroxyl group, the right side of the molecule, and the invisible hydrogen. So this is the first chiral center, which means this carbon is also a chiral center. Now any primary carbons at the end, those are not chiral centers. They have 3 hydrogen atoms, and secondary carbon atoms in the middle that don't have any other substituent attached to them, those are CH2s.
Checkpoint.
And so because they have 2 hydrogen atoms on them, they're not going to be a chiral center. Now what about this carbon? Is that a chiral center? So we have a hydrogen attached to it, an NH2 group, but notice that the right side is the same as the left side. So because the right side is the same as the left side, these 2 groups are equivalent, so it doesn't have 4 different groups. So this is not a chiral center.
Checkpoint.
What happens when light passes through chiral molecules? When plain polarized light passes through chiral molecules, the plane of polarization is rotated, a phenomenon known as optical activity. One enantiomer rotates the light to the right, while the other rotates it to the left. Now let's discuss D and L stereoisomers. These isomers have identical composition but differ in their spatial configuration, specifically at the asymmetric carbon farthest from the aldehyde group. For D sugars, if the hydroxyl OH group on this next to last carbon is on the right, it's a D form. For L sugars, the OH group on this carbon is on the left. It's important to note that the D and L designation does not directly indicate the direction of rotation of the plane polarized light, but rather indicates the optical activity and configuration around a specific chiral center. The examples of D and L glyceraldehyde and D and L glucose clearly illustrate this concept. Here are more examples to solidify your understanding of D and L isomers. We can observe the structural differences between D-fructose and L-fructose, and D-mannose and L-mannose. Notice the mirroring of OH groups on the chiral carbons between D and L forms. Now, let's look at Fischker projections. Fischker projections are a method used to show sugars in their open chain form, primarily for differentiating between L and D carbohydrates. In a Fischker projection of a monosaccharide, for D sugars, the hydrogen on the new ultimate next to last carbon is depicted on the left, and the hydroxyl group on the right. Conversely, for L sugars, the hydrogen is on the right, and the hydroxyl is on the left. So these projections are very useful for quickly identifying stereochemical differences in various diastereomer sugar structures by observing the left or right placement of hydroxyl groups.
Checkpoint.
The structure shown is D-glucose in its Fischker projection. It is an aldohexose or the 6-carbon sugar with an aldehyde group at carbon 1. The hydroxyl group on the nultimate carbon or carbon 5 is on the right, identifying it as a D sugar. The specific arrangement of hydroxyl groups, right, left, right from C to C5 is characteristics of a D-glucose. D sugars, like D-glucose, are the main form of sugar used for energy in living organisms, and D-ribose is essential for building RNA and DNA. D-amino acids are uncommon in humans, but are found in bacterial cell walls, like D-alanine. And some antibiotics, like D-lactic acid, is produced by certain bacteria during fermentation and can be found in fermented foods. L-sugars, such as L-glucose, are rare in nature and cannot be used by human metabolism. L-amino acids are the standard building blocks of nearly all proteins in living things, and L-lactic acid is made in human muscles during intense exercise and is easily converted back to energy by the body. Chirality plays a very important role in the development of pharmaceuticals. It's a fundamental property of handedness in molecules. Although many pharmaceuticals are still marketed as racemates, the body's receptors and enzymes are chiral entities themselves, meaning they interact specifically with chiral drugs. Therefore, controlling the enantiomeric purity and isolating specific enantiomers from chiral drugs is crucial for analytical, clinical, and regulatory purposes, ultimately leading to improved drug safety. A classic example of the importance of chirality in medicine is thalidomide. Originally, it was used to treat morning sickness in pregnant women, but it was withdrawn due to severe effects on fetal development, primarily linked to its S isomer. Researchers later used capillary electrokinetic chromatography to study its enantiomerization. Interestingly, through drug repositioning, finding new users for existing drugs, thalidomide was rediscovered as an effective treatment for leprosy complications and has more recently been proposed for treating various diseases, including cancer, particularly multiple myeloma. Another significant example is levodopa, or L-dopa, which belongs to a class of medications called dopamine agonist. L-dopa is used to increase dopamine concentrations in the treatment of Parkinson's disease, as it is a precursor to dopamine and can cross the blood-brain barrier. L-dopa, specifically the S enantiomer, is a chiral drug with one serogenic center, existing as a pair of enantiomers. The initial use of racemic dopa for Parkinson's treatment led to several adverse effects, like nausea, vomiting, and involuntary movements. However, the use of pure L-dopa, the S enantiomer, significantly reduced the required dose and minimized these adverse effects, as the pharmacological activity resides solely in the S enantiomer, while the R version causes the side effects. Why does the wrong isomer sometimes cause side effects? The wrong isomer sometimes causes side effects because the body's receptors and enzymes are chiral and interact differently with each enantiomer of a drug. While one isomer may produce the desired therapeutic effect, the other may not fit properly or may interact with different targets, leading to unwanted or harmful effects. This is why controlling enantiomeric purity is crucial in pharmaceuticals. Thank you so much for your attention. I hope this presentation has provided you with a clear and comprehensive understanding of isomerism, its various types, and its profound relevance in biochemistry and medicine. Remember, better things for better living through chemistry.